I'm really glad you asked this question because it gives me an opportunity to talk about something that really surprised me, but then I realized that we could help answer. I was struck by the astounding disparity between the severity of the illness that various patients get. Men worse than females, African Americans and Latinos worse than Caucasians, maybe worse than Asians, older versus younger, and then certain disease states versus those without comorbidities. At first it looked as if it was just maybe a sampling error. And then it became very clear this is a prominent phenomenon. And I also recognized that we could help answer this question. The lung organoids, I said, are made from stem cells. The stem cells are made from cells taken from actual patients. And we take these starting cells, whether it's skin or blood, and turn them into something called induced pluripotent stem cells. Now, this is something we've worked with for 10 years, and it's been used to model diseases because of the magic of stem cells, somehow these induced pluripotent stem cells, or called iPSCs, retain a lot of the disease characteristics right in them, so much so that we call them disease in a dish. So cells that come from a patient with Parkinson's disease still retain Parkinson's disease. Same thing with Alzheimer's disease or various other kinds of diseases. Well, we've made the lung organoids from these iPSCs, which means that these are patient-specific lung organoids. And we will be able to see whether, in fact, the lung organoids from a male are different from a female, whether those from African American and Latinos are different from Caucasians, whether young are different from old. But in addition to that, what we can do is we always, as I showed in that earlier slide, start out at the very, very young. That's what turning these stem cells, turning these cells into stem cells does. And we watch development in a dish. And we will be able to see, are the young actually a little less vulnerable because they don't have certain kinds of receptors? But then as they get older, they start developing these receptors, and that's why age makes a difference. We've also learned, for example, that certain environmental toxins, certain perturbagens, vaping, tobacco, seem to accelerate the aging of these cells. And you can imagine that it's more than just vaping and tobacco. It could be the socioeconomic environment of certain groups that accelerate the aging and allow these receptors that the virus glongs onto to have a greater impact. We have the ability of changing the milieu. And this is something that actually has caught the interest of Ben Finley, who's in Christina Vori's lab, who is now interested in exposing these organoids to these kind of environmental toxins to see whether it makes them more susceptible, maybe makes their inflammatory reaction even worse. But there's also a few lessons that we can get from this, just apart from why we have this differential vulnerability. One, if there's something that's going on with the young cells that doesn't exist in the older cells, maybe we can somehow imbue that, either as a drug or as some kind of biologic, to older cells so that they act like younger cells. At worst, what we can make sure is that hopefully we will actually rule out inherent differences. But what we can do is use these to get to the right dose so that when we do get a killer drug, let's say because of what Sumit and his colleagues are going to do, we can make sure, because we will have these mini lungs from many different groups, that either the dosing or the administration compensates for any kind of disparity so that once we do have a drug, it will apply to all comers. What we can also study, because we can manipulate, we can control this environment, is this really, if there are disparities, is it the presence or absence of a particular hormone? If there's a particular disease state, let's say it looks as if diabetics seem to be, for whatever reason, even more susceptible to this disease, we can study, is it a low insulin level or a high glucose in the environment that seems to make these cells more susceptible, and we can learn to compensate that. So I think what we can contribute is, it is in fact that younger patients do seem to be more protected unless they've been exposed to vaping or tobacco. Well, let's understand that older seem to be more vulnerable because they have certain diseases. Maybe we can somehow have an extra kind of therapy for there where we block a particular kind of receptor. So I think that it's intriguing that there's a difference that there's this disparity. It's perturbing. But our job is to try to compensate for it so that when we do get the best cocktail of drugs, it will be useful for all comers, regardless of age, of comorbidities, of racial background, of gender.